Alrighty. Well, hi everyone. We are the Lemurs and Enrichment group. Our group is made up of myself, Mallory, Dailey, Faye, Ethan, Samantha, and Harry. So 91% of all lemur species are classified as either critically endangered, endangered, or vulnerable, according to the IUCN Red List. Because of that, many zoos house lemurs to promote their conservation and to act as animal ambassadors for their species, and basically to promote their conservation back in their native range in Madagascar. Primates in zoos provide researchers a unique opportunity to detect valuable behavior research and allows observers to detect behaviors that might not typically be available to them in the wild. And not only are lemurs in zoos very valuable to researchers, but they're also a great attraction to many visitors that come and visit zoos. They're probably one of the most popular species that people come across. They're exposed to a variety of food items. They interact with guests and visitors. Their enclosures change fairly regularly, and they receive a lot of different types of enrichment items. And so there are over 100 species of lemurs that are all found in Madagascar. And lemurs, like I mentioned before, they're the most threatened group of mammals in the entire world. Madagascar is facing a lot of conservation issues such as deforestation and overhunting, which is causing a lot of issues for the lemurs. And so we decided to focus on 3 species. The 3 species were the ring-tailed lemur, the red ruffed lemur, and the black and white ruffed lemur. And we chose these 3 species because of their availability in zoos, and they're basically the ones that group members had access to. And so ring-tailed lemurs, they're probably the most popular species of lemurs that are found in zoos. There's close to 3,400 lemurs ring-tailed lemurs in zoos as of 2014. And these ones are a little bit unique compared to other species because they are actually the most terrestrial in ground foraging of all lemurs. And they also are kind of more found in the southern part of Madagascar that's a little bit more arid and dry. But these guys are very vocal. They form large social groups, and they're very adaptable to living life in zoos, which is why they are so popular there. The next species is the red ruffed lemur, and these guys are critically endangered, and they're only found in that tiny little area in the northern tip of Madagascar. And what makes these guys a little bit unique is they're one of the largest species of lemurs, and they're also the most fruit-eating of all lemurs. And so they're very highly motivated from fruit and food. And they also tend to live more in the canopies of the trees, which is why they have issues, especially with the deforestation issues that are going on in Madagascar. And then the last species of lemur is very closely related to the red ruffed lemur. This is the black and white ruffed lemur. This is one of the largest pollinators and is also one of the largest species of lemurs that are found. And they also, like their cousin, the red ruffed lemur, prefer to live in the upper half of canopies.
So to get into enrichment, enrichment is defined as the process for improving or enhancing zoo animal environments and care. Typically, it involves the identification and subsequent addition to the zoo environment of a specific stimulus that was not previously present. Enrichment is extremely important to highly intelligent organisms like lemurs. These events promote curiosity, exploration, and stimulation to keep individuals from being bored. So enrichment comes in many different forms with one common goal. And some examples include, but are not limited to, changing the structures with their enclosures, training individuals to do various tasks like step on a scale, introducing new smells, toys, or visual stimulation devices, and even finger painting and puzzles. These methods are often created by the people who care for the individuals at the various zoos and different facilities they live at. And it's much more difficult than just throwing in a toy or whatnot to the enclosure. There's a specific goal in mind of these individuals, and that's what they use to create these devices. And that might be creative behavior that they would display in the wild. So As previously mentioned, enrichment comes in many different forms, although one goal is common throughout. The methods of enrichment are different. Feeding enrichment includes the manipulation of food or method of feeding. Tactile enrichment includes providing an object or a toy. Structural enrichment includes the alteration of the enclosure. Visual enrichment includes the addition of a visual stimuli. And olfactory enrichment includes the addition of some sort of odor. And it's important to mention that a single enrichment device can involve multiple of these categories. So that gets into the purpose of our project. Enrichment's effective at reducing stress of captive individuals, which has reproductive, immunological, developmental, and behavioral implications. However, providing the same methods of enrichment may become boring for individuals and defeat their intended purpose. That's why increasing the understanding in regards to what methods are stimulating individuals the most and providing the most amount of benefits is an important piece of developing new enrichment ideas. A study was done by Taylor Shire of Iowa State in 2012 describing the differences of captive and wild ring-tailed lemurs. He found that captive individuals were less active and they did not display the typical sunning behavior of wild individuals. And this is a really important behavior because it's something they use for thermoregulation. So when you're developing a new enrichment device, you might look to take Taylor's research and ours and kind of put them together and figure out what enrichment method works best, what are individuals most stimulated by and spending the most time with, and what are the goals of it? Are you trying to promote activity? Are you trying to promote a wild behavior like sunning? And in doing that, you can kind of put those together and provide an opportunity for a really endangered species that promotes wild behavior to maybe for the point of, reintroducing these individuals if the populations become so endangered at one point, which in my opinion, they would look to the most active and the ones that display the most amount of natural behavior, like the sunning.
Okay. So to provide some more insights to help optimize enrichment processes and to improve interactions, we asked the central question that's up on the top right there. How do enrichment type and species impact the duration of interactions with enrichment devices in zoo lemurs? And so our first hypothesis was individuals will interact with feeding enrichment for a longer average duration than with environmental enrichment. And environmental enrichment is both structural and tactile. Our second hypothesis was that ring-tailed lemurs will interact with enrichment devices for a longer average total duration than ruffed, the red ruffed or black and white ruffed lemurs. There were some variables, the role of some variables like sex and age, we could not really explore because, like we wanted to, because data on those variables was not always available. So here is just a visual to help illustrate the relationships and the variables that we tested. The big circles are our independent variables and the little circles are our dependent variables. And so we wanted to understand impacts on lemurs' visual interaction time with objects, their tactile interaction time, and the total interaction time with objects. And total interaction time was just the visual time plus the tactile time. So we gathered our data and made behavior observations at 10 different zoo venues, zoos that house lemurs and that provide enrichment. Some of that data we gathered in person, but due to COVID restrictions, much of that data had to be collected from either live Zoom, live stream zoo video or recorded video. So among the data that we collected was data on the subjects and data on study sites. So we recorded facility location for where the observations were taking place. We recorded the date and time of the observations, the species of individual being observed, the sex of the individual and the age of the individual if we could, Species was very easy to determine by appearance, but sex and age were hard to determine unless caretakers specifically provided that information. So also among the data we collected was information on data on enrichment type and behavior data. So again, enrichment type was either feeding or environmental. We recorded the duration of tactile interaction, the duration of visual interaction, total duration of interaction, and we matched that data with its corresponding subject individual. In defining our categories of behavior, we defined tactile interaction as any action where the individual is in physical contact with the object of enrichment and or is consuming food from the feeding enrichment device. Visual interaction includes looking at and or approaching the object without making physical contact. In measuring the interaction durations, we tried to make our in-person observation sessions and our live stream sessions and our recorded video sessions as similar as possible. So the time period for collecting data, if we were doing an in-person observation or a live stream, was 10 minutes from the onset, the installation of the enrichment device. If we were looking at pre-recorded content, we would observe for the entire length of the video unless it exceeded 10 minutes, in which case we would stop at 10 minutes. And so we recorded our times using a stopwatch or a cell phone timer. I think everyone probably used cell phone timers. And so also in measuring interactions, for visual interactions, we started timing at the first site of the enrichment device by the animal and continued recording as visual attention was retained, including if the individual approached the object. And we stopped timing once the enrichment device lost the visual attention of the individual, or if the individual transitioned to tactile interaction. So tactile interactions, with tactile interactions, we started the timer at the first touch of the object by the individual and continued recording while the individual remained in contact or held or consumed food. We ended the timer when the animal was no longer in physical contact with the enrichment device or was no longer consuming food from the device. And so we ended up with subject data and interaction data for 53 individuals. And it was from this data set that we made our statistical analyses. So I think we'll transition to Faye, who will discuss results and analyses.
Yeah, so just as that slide exactly says, we had raw data for about 53 individuals and 60 data entries from that. So this data needed to be corrected because, as mentioned, we were observing independently of one another for the most part. So some observers did utilize the same video. So we needed to correct that for observations of the same individual, as well as observations where individuals interacted separately on more than one occasion. So for each of those cases, an average was taken, and our final sample size was 34 data entries corresponding to 34 unique individuals. Okay, so of the total observation period, between every interaction observed or every video or live stream or in-person attendance, 30% of the time we actually observed interaction by the lemurs with the enrichment. And of that time where interaction was observed, there is almost a 50-50 split between visual and tactile interaction. When we ran our t-test analysis using the Microsoft Excel data analysis program, we found that there was statistical difference to support our first hypothesis of the differentiation of duration of interaction between feeding versus structural enrichment. However, this statistical difference was only found for the tactile interaction and the duration of total interaction. The difference was not found between feeding or structural enrichment for visual interaction. And then as you see in the next slide, for the difference between the species, there was no significant difference at all. So our second hypothesis was not supported by our results. We found no significant difference for the duration of visual, tactile, or total interaction between either of the enrichment types. So when we looked at the distributions for all of our data for the interactions, we saw that in almost every scenario, there is a right skew. And this was caused by individuals that interacted for a longer period of time than the other individuals. So we had outliers interacting for a large amount of time. That was mostly concerning feeding enrichment, because as mentioned, we did continue recording tactile interaction while the individual was consuming food. So that was a main cause of why our distributions are skewed to the right. As you can see in this figure, when we plotted the line graph for each duration of visual and tactile interaction for each individual, we saw a pretty clear pattern in most of the cases where there was a large discrepancy between the visual and tactile interaction for any single individual. So for most cases, as you can see, if you click for the first arrow and the second arrow, if they come up, if you click, yep. For most of the cases, there was a large discrepancy, whereas if the individual was interacting for a very short amount of time visually, that was followed by a long duration of tactile interaction, or vice versa. As you can see by the circle on the graph, there were not as many cases where the visual and tactile duration of interaction aligned or were similar, unless the individual did not interact at all. So that was another correlation that we saw, but since it was not observed in every individual, we could not draw statistical significance, but it was another correlation that we observed. And as mentioned, although we did not have a large enough sample size to draw any significance from the differentiation between male and female interaction or juvenile and adult interaction, although we found a significant difference between males and females in the duration of their tactile interaction, our sample size was not large enough. So these results may be different had we had a larger sample size, and maybe something that we would consider trying to gather more data on in the future.
So based on our observations, we conclude that resources should be dedicated to feeding enrichment over environmental enrichment. We looked at tactile and visual interactions, and as discussed earlier, the total duration of the interaction was longer for an enrichment involving food. Food is a driving force for the lemurs, which leads to more physical and mental stimulation if they receive a reward for these interactions. Lemurs still need other forms of enrichment, though feeding enrichment is most successful at improving the lemur's quality of life in captivity. In the bottom right here, you see I have inserted some photos of a couple lemurs at the Tennessee Aquarium. Since I work here, I was able to come into contact with the keeper over this area and obtain identification sheets and some other background information about the individuals at the Tennessee Aquarium. So the first photo on the left here is Yoda. He is a male ring-tailed lemur and is 5 years old. Then in the middle is a red ruff female named Josephine. She is the oldest lemur at the Tennessee Aquarium at 20 years old. And lastly, on the right is Gil, which is a 6-year-old ring-tailed male. They have more lemurs, but I thought it would be fun to share a couple photos of the feeding. As you can see here on the right, which it's actually covered up by our faces, Gil is actually interacting with some feeding enrichment right there. So some improvements which would have enabled better observation is in-person data collection for the entire project, rather than live streams, recordings, and in-person visits. Though this was challenging, given that we are an online program and in-person resources were not available to everyone. Along with this, more background information regarding the sex, age, and personality, which is really challenging to see from just simply viewing the individuals, would have enabled us to make more specific hypotheses and interpret the data in more depth. An example of this would be comparing the sexes, age groups, and the species. Also, having a larger sample size would allow these factors to be more dependable to the hypothesis. As there was 34 individuals and 60 data entries, increasing these numbers would aid in this. Though due to the COVID-19 pandemic, adequate social distancing, facility closures, and limited guest count, it presented difficulties for these observations. After the pandemic has concluded, more reliable and specific data could be collected. And then lastly, we have our publications that we used. And then we have the website publications as well, or the citations.